How's the inquisitive going? We got through, I changed the setting now, so you should be able, it should update your scores automatically. So I think, so just let me know if you think I went back and changed the chapter one for most people. So let me know if I missed you or if you're having issues getting into the system, come into my office hours today so I can show you how to get onto Canvas and how to do all that, okay? So is it working out for the rest of you though? Is it about half an hour or so to do it? That's the time setting I put on there. So I didn't want it to be too onerous, right? It's there to try to introduce you to the material. So doing it early on in the chapter, whatever chapter it is probably beneficial as well. Then you can go back and review if you need to review. Nothing? Okay, so today we're going to talk a little bit about microscopes and microscopy. And then on Friday, we're going to start a little bit of a review of Gen Bio. So you may want to go back and start your review of Gen Bio, macromolecules and things like that. You will have a quiz early next week. So that'll be on Canvas. That'll give you an idea of some of the questions that I expect you to know on the test. That's in mid-September sometime, okay? So just beware of what's coming down the pipe, okay? And then I think for labs next week, we're doing the epidemiology labs, right? So take a look at those and figure out, see what we're gonna do, so. Okay, so today we're gonna talk a little bit about what is microscopy, what kind of microscopy do we have, how do we get images of it, and then of course, why do we care? Why do we care? What does it allow us to do for microbiology, obviously, because that's what our class is. Some of these lecture slides are pretty nitty gritty. They'll show you like the physics of light and that kind of stuff. I'm not going to test you on the physics part. You can take a physics class and do it that way, okay? But there's some interesting things we can learn. And some of these things will help you when you start using microscopes in lab. They'll help you use them correctly and then hopefully avoid issues where, you know, I see students focusing in on a piece of fuzz or dust or something like that, and you can figure out that you're not seeing what you're supposed to be seeing, and so you can adjust things for yourself. So that's why I included a microscopy slide or a lecture in the slides. And of course, it's useful, right? So there's a little case study in here where some kids got sick from canned foods. And so, of course, they had a certain symptoms of paralysis and sickness. And so the doctors have to go through, okay, what could be the options here? And they focused in on botulinum. The botulism is one of those ones. Didn't we already talk about this? I think we already talked about this a little bit, but it's one where it produces a toxin that produces paralysis. And it's common in canned food. So I think when the doctors heard that maybe it could be a foodborne illness and related, then they could focus in on that using the symptoms. So what do? They traced it back to cans of chili sauce. And so then they tried to isolate microbes out of there. And here, using a stain and the microscope, they were pretty quickly able to focus in on a particular type of organism. And why this one was why it stood out so much is that this was an endospore stain. So you could see the endospores. We'll talk about what those are. But spores are basically like a seed, and they're very resistant to either killing or drying out or things like that. So it's a common way for bacteria to survive under non-optimal conditions. And then when conditions are right, then they turn back into what's called a vegetative cell. And so that's what happened here. The spores live just fine in the cans. So for some reason, the canned foods were not properly treated, whatever it was. And then those spores were allowed to stay in there. And then when they were ingested, then they were able to come back to life essentially, and then start producing the toxins. So that's what produced the symptoms. So I mean, when you can get really distinctive cell structures and cell types under the microscope, and that can help you very quickly identify what you're looking at. So that's really the point of this whole class today, is to convince you of that and kind of see how they work and how it helps us, right? So there's some terms Most things that we're looking at are microscopic, so they're too small to see with the naked eye. We could barely see some of the largest eukaryotic cells, but most bacteria are under the threshold or the limit of what we can see. So of course, we use microscopes to magnify and resolve these structures. And it's going to help because most bacteria are smaller than red blood cells. So you can do in the medical lab, you can do what are called differential cell counts. So you can spread blood out on a slide and actually see those cells pretty well because they're pretty large. But most of the bacteria things we're looking at are smaller than that, except for some of the larger things like trypanosomes. Obviously, those are a little easier to see. So again, this kind of relates back. What did we talk about in terms of the germ theory? How did microscopes help us fast forward the germ theory? See somebody over here. It's from our history lesson. Just so I don't come over there. Give these guys a chance. Huh?
Maybe microbes visible.
Yeah, right.
And they could see like they're washing their hands and how well like they did or did not wash them.
Did they put their hands under the microscopes? I don't know. That's a good question. Well, they could have washed it off though, right? So it was basically being able to visualize, that's the word. Visualize these things that were associated with disease, right? So Leeuwenhoek, right? Or Leeuwenhoek, however you say his name. He was able to let people see these things that they actually were there. Right. So that's why it was really, really cool. Okay, what are the different types? The truth is we're only going to use one type in the lab, light microscopy, okay? So, but there's lots of different types depending on what you're trying to do. So light microscopy, you can either use stained or unstained organisms or whatever your cells, whatever you're looking at. And most of them are going to absorb light and look dark against a bright background. This is light microscopy. Dark microscopy is the opposite. And I'll have a slide later at the end, which shows a dark background and the specimens kind of reflect light. So they look whitish against a dark background. Okay. So that's pretty much the only one we can use. It has a lot of limitations. As you'll see, you don't see a lot of cell structure. So when we start looking at things, you're not going to see a lot of the internal structures, unfortunately. So you might, at 100x, when we use the 100x objective and oil, you're going to barely see bacteria on there. And all we can do really is look at shape and size and morphology, those kind of things. But there's other cool things. You can use phase contrast, which when you change contrast, that helps your eyes differentiate the structures better. You can use fluorescence. I don't know if I spelled it wrong up here, but I hate English. So fluorescence is basically where you usually treat the cells with some sort of fluorescent molecule, and then that fluorescence helps you visualize them under the microscope. And then for the tiniest things, you can use electron microscopy. We actually do have an electron microscope in this building. It's in the basement. SC. I don't know if anybody ever used it. Anybody work with Chris Monson? You guys, I don't know if you do chemistry. So maybe wrong crowd. But we have different types of electron microscopy, and those processes are going to be for the smallest types of structures. So we're not going to use it in this class. So with light microscopy, we can easily see the largest organisms. So this is a eukaryote. Actually, they're both eukaryotes. So you can kind of see what the structures are. You can stain them or not, depending on what you're looking at. And the bacteria is so cool. And microbes are so cool. You can see all sorts of different cell structures. Look at that. I mean, isn't that cool? So we're going to do some gram staining. That's probably the most well-known differential stains. Is what we're going to use. We do gram staining, but just look at the one of my favorite words, the plethora of different cell structures that we can see. You guys know that word? The plethora of piñatas. You ever watch the Three Amigos? I'm dating myself. That's like 80s, isn't it? 80s. Okay, never mind. It's a plethora, right, of wide variety of cell structures from just simple rods to spirili to all sorts of different cool things, right? And you guys undoubtedly have seen pictures, maybe when Ebola came out. Ebola virus has a cool structure that they showed when they first identified it. So again, we can use this quite easily to get information about what the cells look like and what they look like. That's what I was saying. Here again, this is repeated. So light microscopy is for the largest structures. Then here, this is an E. coli in the middle where you can start seeing the internal structures, but you have to use electron microscopy for that, really. So, I mean, for all that's cool about it, it's light microscopy is pretty limited to the largest structures, okay? Let's see, other things, I'll just mention X-ray crystallography, it might show up. This is not really microscopy, but you bombard samples with X-rays, and the X-rays actually scatter, and then you analyze the scatter pattern from that to get an idea of structures. So people have used that to look at DNA, ribosomes. Really, we're talking about the smallest structures that you can't really see ever by eye, okay? Kind of cool. So what do you need to know? Probably what are the types? The ones that we're going to use the most are bright field, and just be familiar with what's available out there, right? So far, so good. Okay. I have some questions for you later. Okay, properties of light. So I'm just going to kind of quickly go through this. You should remember that light is a wave and a particle. The wavelengths have to be relatively small and shorter compared to whatever structure you're looking at, because that's what's going to dictate if the waves actually interact with the particular structure, whatever you're looking at, okay? Let's see, there's a couple of different things that can happen when light interacts with these objects. And I'll teach you when we're in lab, or hopefully whoever's your lab professor, they'll show you how to adjust the contrast on these microscopes to adjust just the perfect amount of light that interacts with whatever organism that you're looking at. So the microscopes are kind of cool in how they can work with whatever sample you're looking at, as long as it's big enough, I suppose. A couple things. So of course, light can be absorbed. We're not really worried about that. Really what we're looking at is light that either refracts or reflects or goes through a sample, is transmitted through a sample. Some of this we'll have to deal with when we use the 100x, because of the way that light interacts between glass and air, called the refractive index. It actually changes direction. So we have to correct for that when we use some of the microscopes in the lab. So it kind of just shows you what's going on with those. I'm not going to test you on the difference between reflection and refraction and all that, okay? So you can kind of just breeze through this and try to get an idea of it. This is what I was talking about with the smallest samples. So when we use the 100x, we're actually going to have to use oil on top of our glass slides. And that's going to help us correct for this refraction that occurs when the light goes through glass. And that's just because wavelengths, they act differently between air and between glass. So there's some theory behind this. There's a shift. Again, I'm not going to really test you on it, but I want you to know why we have to do that, okay? So you'll see in the lab, we'll start with like the 10 or 4x, 10x, 40x objectives. We'll talk about that in a second. But then on the 100x, you'll notice that we have to use an oil to deal with this refractive properties of the images, okay? Let's see. So when you take physics, you'll learn about focal points and all that. So as light goes through these lenses, there's typically a focal point or focal plane. And we're usually focusing in on the focal points of where the light is actually going to interact. Interestingly enough, when we look at structures under the microscope, they're going to be inverted because of the way that the light deals with these light microscopes, or sorry, lenses, when they go through the light. microscopes. Okay. Let's see. Let's throw a question at you guys here and wake you up. How about that? Okay, this is an easy one. Throw you a softball. Oh, I gotta get used to the fact that I have to do work with these flicker things. I can't just make you guys do the work. So what is the best microscope to see bacteria? Bacteria. What am I doing here? Oops. Press the button. So remember, when you see your name appear, then you can put your card down. Hopefully I have everybody's clickers numbers at this point. Your hand is- Does it come up? Okay, great. Folks, can you email me your name and people?
Yeah, I did.
Well, I must have screwed up, sorry. Okay, did I get everybody? Okay, cool. So what is let's see if show the correct answer. What is electron microscopy good for? What kind of? Yeah, right, yeah. So when you want to try to start seeing organelles within a cell, that's when you start using electron microscopy. What about fluorescence? We haven't talked about it much yet, but what would that be? I did mention that you have to use some sort of a probe or something that fluoresces, right? So what do you think you could use that for? Take a guess.
Cancer treatment, or just like, I'm forgetting the name of it, a specific name,
Yeah.
It's just by detecting if there's cancer present in the cells. It's like fluorescent dyeing, I'm pretty sure.
Right, and so what you'll learn is that dye, those fluorescent things will target specific things. So with cancer, you can use dyes that they know will stick to a particular cancer. Like what do you want to call it, an ID or a protein or something that's specific for that. So you, not just cancer, right? You can use it for basically anything. So if you want to look at a particular organelle, you can actually label organelles like I use it for chloroplasts, I'm saying chloroplasts. So you have probes for chloroplasts and they'll fluoresce the chloroplasts. What's none of the above good for? Nothing. Okay, so light microscopy is good. What is that good for?
Bigger structure.
Yeah. So bigger things, think about like human blood cells. Those are pretty easy to see. And then smaller microbes, you can just barely make them out. Okay. Okay, questions on that? That was an easy one. I have one more, but we can do it. It kind of goes along with this slide here, but I'll come back to it. Okay, bright field. So with bright field, you have to think about wavelengths, contrast, and lens quality. Of course, we don't have the top of the line microscopes here at the university, and they tend to get beat up a little bit, so we have to be nice to them and talk nicely and clean off the objectives. So when you're using microscopes in the lab, make sure you're following directions and storing them correctly and cleaning them correctly, especially since we're going to go back and forth between the oil immersion lenses and the regular ones. Okay, it's going to take some concentration. But obviously with the lenses that we do use, we want to hopefully have pretty good resolution between those lenses. So we'll see that with the lower power magnifications, the resolution is not as good as the higher powers. That's why we always want to try to get up to the highest power we can, depending on what we're trying to do, I guess. But usually that is going to allow us to see things a lot better. And those are the different objectives. So actually, where's that picture? I'll skip ahead here. There's a picture of a microscope here. So the different things we have, you have the eyepieces. also called the ocular lenses. So here, this one, our microscopes have two ocular lenses. Those have a magnification, 10x magnification, okay? So obviously those are the things that you're gonna look through. And then the objective lenses, they're down here. So I usually call it like a nose or something. Okay, and you can churn it around. You guys remember using this in Gen Bio at all? Maybe not, okay. So hope some of you might have been exposed to the microscopes, but you'll churn the objectives and you always start. I know I'm getting into a little lab stuff here, but that's okay. You always start on the 4x, 10x, and 40x. You always start with the lowest magnification. And then the focal knobs are not going to be at the top. They're actually going to be on the bottom. Sorry, I should have put in a picture of the actual microscopes that we use. Now I know I'll go back and change it. Okay. So you'll have the focal knobs, you'll have a coarse focus and a fine focus. Okay. So you'll focus on that. What's cool with these microscopes that we use is it's called parafocal. So in theory, when you focus on the 4x and then you change the 10x, you shouldn't have to change your focal plane very much at all. You should only just have to barely touch the fine focus. But here's where, you know, the microscopes don't always work the best. So you'll probably, more likely than not, you'll run into a situation where you can't get something really focused very well. So you have to play around with it a little bit more or clean off the objective lenses. We always use a special cleaner and special tissue paper to clean off the objectives. So you always use lens paper when you're cleaning off any of the lenses in the microscope, not paper towels. Here. So the objectives, that's really where you get the resolution to look at whatever sample that you're looking at, okay? So that's where that comes in. Now there's other things that you can use. There's a condenser and a diaphragm. The condenser here is below the stage. So the stage is where you put the lens, not the lens, the slide, the microscope slide with whatever sample you have. I do have a pointer. So you have a stage and then the slide. Below that is the condenser. The condenser is what's going to condense the light. So you have a light source down here and it allows the light to come up through the sample up to your eye. The condenser collects that into a column and then you have a diaphragm. That works just like the, what's that thing on your eye that opens and closes? The iris, thank you. Sheesh. Sorry. It's just like the iris. So it will change basically the diameter of light that goes through. And what does that help your eye with? That actually helps your eye with contrast. And so this will help you with contrast as well. So usually, sometimes I'll come around and you can't see a sample very well. So you close down that diaphragm and then it helps you actually increase the contrast of whatever sample you're looking at. So it's pretty cool they have there, okay? So I'm giving you a preview of your lesson in the lab of what you're going to do with these, okay? And you're going to get good enough to use them where you can start troubleshooting what's going on. Any questions on the different types or different parts of the microscope? I suppose I could throw something in on a quiz or test on this, but it's more of a lab topic, so probably wouldn't focus on the parts of a microscope in lecture, but it is fair game. We did talk about it in lecture. So questions on that at all? No? Okay. Isn't it cool? Microscopes are cool. Yes, it's cool and it can help us see things, right? And identify them. Is it important to get things focused? Actually, that's a question. Here we go. Here's another question. Come on. Make up for this here. Whoops, I can advance it on here. Is it important to have your sample in focus? Okay. So sometimes I ask really dumb questions. That's okay. Right? Okay. I think you're covering this.
It.
Oh, or not, but
Oh,
on by. No, I think we just have to. You just have to do this enough that you guys, it's just second nature. Do you get when you get a finger over it, then it doesn't seem to like it. But it's kind of picky, is that the right word? Did I get everybody? The word wolf fixers, sorry. It's always gonna be. So there's always that one person, yeah, who says no. Yes, it is important to have your things in focus. If you don't have it in focus, then it's going to be really hard to see what's going on. So in this sample here, you can see things in and out of the focal plane, in and out of the focal plane. So this will take some practice. When you're in lab, you're going to practice setting up the microscope, making sure you're actually looking at what you're supposed to be looking at, and then focusing so that you can see a nice sharp image. And if you see things that are out of focus, sometimes they look empty, or they're blurred, or if they're actually, if you have and this is what's called a wet mount. So this is where you put liquid onto the slide and then you put a cover slip over it. Sometimes you can have things in different focal plane. So you just have to kind of practice looking at it to see where the correct focal plane is, so you can see your samples. So I think the lab is always more fun than lecture, isn't it? Usually, yeah, it is. Because here I just blab for a while, but in lab you actually get in and get to do things, so it's pretty cool. Okay, how are we doing? Any questions? Questions on expectations so far? You guys are quiet today. What's wrong? We shouldn't be tired yet. We have like 13 weeks left. It sounds bad. Nothing? Okay. Well, just let me know if now I throw some softball questions at you, but really I do expect you now to kind of know what types we're talking about, what type we're going to use the most, how it's useful, how it might be lacking, right? Those are the expectations so far. No, I'm not going to concentrate on physics of light on the test. Okay, that's a different class. So you could take a physics class. Okay. Okay, let's talk a little bit about staining. Here's another case study. So a young child gets sick, and I think this is in India, right? Somewhere, okay? And they have a fever, cycling fevers, and they're very tired, okay? So what they find is they find this organism called a plasmodium in their blood. So here what they do is they take a blood smear, so a wet mount, and they smear it out onto the slide and then put a cover slip over it. And then you add some sort of a stain, and this is what's called a GMSA stain. GMSA, I think, stains nucleic acids mostly, if I remember correctly. So it's almost like a fluorescent thing. It binds to structure specifically, but it's not a fluorescent dye. It's basically what they call a differential dye. So you can see things in the cell. Look at, isn't this kind of creepy? So Plasmodium is one of those organisms that's intracellular. So it gets into cells at least at one point in its life cycle. This one has a couple of different stages of life cycle, which we'll probably cover at some point. This one during this stage shows up in the blood cells themselves. So that's kind of creepy. Okay. And this causes malaria. And it's transmitted through biting flies, right? Kind of cool. So fixation and staining, this is another thing that we'll do, is we will take bacterial, usually bacterial samples or eukaryotes. You add them to a little drop of water on a slide. So if you have your slide here, you put a little drop of water on the slide, and then you're going to put a cover slip over that. And then what we'll do is you could take samples either from liquid or from Petri dishes. You could take samples of the bacterial cells, you put them into the water, and then you kind of spread them out onto the slide. So again, a version of a wet mount basically, except here, we're going to usually heat fix them first. So you don't just hold it over a flame, because then they'll just splatter all over the place. Okay, you'll learn this in lab. But you heat them a little bit first to fix them to the slide, and that usually kills them. And then you apply stains on top of that. And then we'll do a couple of different stains. We'll do a simple stain, and then we'll do a gram stain in the lab. And the stains will help us see the cells better, okay? So here, this is the kind of the steps I just talked about. So you put the sample on there, kind of heat them or air dry, and then you're gonna go through the staining process. And then sometimes you put a cover slip on there, sometimes you don't need to. Other times you can just take the slide and look at them directly, okay? So what do stains do for us? What do you think? There's a couple things.
They add contrast.
Add contrast and kills the cell. Could it stain particular structures? Probably a third. Yeah, so sometimes it stains particular structures, sometimes it doesn't. This one, this is a gram stain. So this one doesn't stain nuclei very well, but you'll see in some others that you'll see that we do. Yeah, like methylene blue, the nuclei will actually be a little darker than the surrounding cells. So sometimes stains can stain different structures differently. But here, this one is showing there are these are cheek cells with a bacterium. So it's from a sputum sample, a spit sample. And then this one, probably a gram stain. So gonna either show up pink or purple. Okay. So right, we can see cells better. We're gonna learn that for something like a gram positive, gram negative, that will actually classify the bacteria based on cell wall structure. So we're gonna talk more about this later on. But using a gram stain can actually tell us what type of bacteria that we're dealing with. Not the strain exactly, but the class of bacteria. And this can come into play. So when we talk about antibiotics, sometimes they're more effective against gram positive than gram negative, or vice versa. So it depends. If you can figure out what kind of organism you're working with as soon as possible, you can start a much more effective treatment as soon as possible. So that's what the staining is really useful for. So we will talk a lot more about gram stains, how they're useful, and we'll actually do them in lab. So it's kind of fun. And just as an early reminder, anytime we're doing staining in lab, don't wear your favorite clothes. Okay? Because stains are staining. So they can stain clothes. Sometimes if you get them on your hands, they'll be there for a few days. They won't kill you, but you'll have purple fingers or something. But we have gloves in lab. We'll hand out lab coats next week as well. So that'll help protect you a little bit. So, but gram stains are kind of fun. And it's kind of exciting. We'll use that as part of our identification process when you do your unknown. Sometimes cells don't have cell walls, like Mycobacterium. They don't have a true cell wall, so you have to use other kind of stains like acid fast, or what we started with at the beginning, an endospore stain. So you can, again, stain particular structures to help you see what is going on. Okay. Kind of cool. I don't know. I always like looking at pretty pictures, don't you guys? Okay, other types. So this is just for FYI, not really going to test you on it, but I did put bright field versus dark up here. So that might show up in the notes or in the book. Okay. So here's an example of a dark field. What's cool about this one is instead of letting light go directly through the center of the objective, they actually have a disc in the middle that blocks the light. So the light kind of goes around the sample you're looking at. So what you get then is a dark background, and then whatever light gets around it is scattered by the sample. So this is a I don't know how to say that, spirochete or something. So it's a spiral shaped organism. And it is scattered by the light that goes around the sample. Kind of interesting that way. And this helps, if you don't have very good contrast with bright field, can't see them very well. Sometimes the dark field things will show up a little bit better. Phase contrast is a little trickier because you're working with two different wavelengths. So again, I just focus on the name. Basically, this is used to help us really increase the contrast of whatever sample that we're looking at, okay? So again, I'm not going to test you on this, but you can kind of see what's possible with these kind of samples. And another case or another example, interference microscopy. So this one again gives you kind of a 3D view of whatever structure you're looking at. Cool. Oh, some of you might go into microscopy or something, right? Or a medical lab. You can play around with all sorts of microscopes. Kind of cool. Okay, I have one more case study in here from a hot tub. Think about hot tubs, they can get kind of gross if you've ever had one, right? Nice hot conditions and moist, right? And then there's usually dirty humans that get in there and introduce all sorts of things, right? Yeah, and of course, if you don't treat the water every once in a while, then it can get even worse, right? Anybody have that experience? Nobody wants to fess up? Okay, yeah, okay. So here, somebody gets sick from, or at least after being in a sauna, kind of interesting, with a flushed membrane, what is it? I guess it's a fever and dry mucous membranes. So he shows pneumonia and C-reactive protein. So that's related to immune response there, okay? So he talked about cleaning a filter on a hot tub and could have been contaminated by something. So what do they do here? They use a fluorescence. So they can use a fluorescent stain, or this one is, you can also do a gram stain, and it identifies a particular organism from that, okay? So here again is an example where you can use a particular probe to bind to particular structures to see what you're kind of looking at. And here, I think in one picture, they're actually looking at the expression of a particular protein. So we'll learn on Friday that proteins are very, very important in the cell, and you can learn a lot about a bacterium or a microbe based on the proteins that they have inside the cells. So that's kind of cool. That's what some of those cancer cells look like, don't they? You can get some really cool pictures of cells, and you can actually identify particular structures within the cell. It's kind of cool. Okay, and I think this is the last one, right? Just some electron micrographs. So here with electron micrographs, you can see the smallest structures. Smallest structures. What is that? That's actually a bacterium. Here you can actually see ribosomes. Not very well still, but you can actually kind of see the structure of them. Those are the smallest things there. Okay, so any questions on today's material? In terms of expectations, I tried to give you some hints about what I care about, and what I don't care about.
Can you just say again which types you want us to focus on most?
Well, probably bright field and dark field. Those are like, the only one we're gonna use in lab is bright field, so. There's really only one. And it's very easy to use and very useful for bacterial types. But it does have some drawbacks. So, I could ask you, what's the best to look at bacteria? Now, and there could be more than one answer, right? So either bright field or electron microscopy, right? Then if I said, what if you wanted to look at ribosomes in bacteria? Then he would say, well, definitely electron microscopy. Is that okay? So from lab, you'll learn the parts of the microscope. You'll know what eyepieces are and objective lenses. So they could show up on a test, but it's really not going to be an emphasis. for the lecture.